Welcome to Biopsychology, where we'll explore how our thoughts and behavior are influenced by our bodily systems, our nervous system, and even our brain. Just to give you a preview for what we'll cover in this slideshow, we're talking about the biopsychology perspective as another explanation for things we want to explain. This is a relatively new area compared to some of these traditional perspectives such as the humanistic and behavioral. And we know so much now about biopsych that we really can't ignore it, even if we're more oriented toward other perspectives. In this slideshow, we're going to cover a little genetic terminology. We'll introduce how neurotransmitters and neurons work, and also some neural systems within the body. But first, some true-false. As you might imagine, we do lose neurons every day. There's some debate about the actual number, but it is far less than 100,000. Best estimates that I recently saw were about 10,000. Think about that. Every day you lose 10,000 neurons or so, and you don't wake up the next day feeling any dumber, which goes to show you first just the sheer number in billions of neurons that we have, how long it would take to lose all of them, and also the many redundancies that the system is capable of if there's damage in one place or another. Here is a common statement that is actually a myth. Drinking alcohol, even heavily, does not appear to kill brain cells. It does appear to damage the connections between neural cells. So the cell still lives, the neuron still lives, even under heavy alcohol abuse, but it does impair brain function to some extent. Here's a common myth as well. Seems like there's a lot of belief about this 10% myth. It's really permeated throughout the world, and it's very, very much untrue. We need to use all of our brain, and we'll see how it develops in a very precise order normally so that each piece of real estate in our brain is used efficiently. This used to be commonly thought to believe to be false, when actually it's true, although I want to be very precise. This is only true in a few documented areas. So generally, if we do have brain damage, in most things, we don't have the option of growing new neurons, but there is some cases where humans have the capability to do it. And I think we're going to learn more about this in the future. This was a common belief that people in a coma could be awoken by exposing them to things they're familiar with. This is false. Of course, depending on the severity of the coma, there is less and less that a person can be responsive to. Another key thing to keep in mind is that this is definitely false. It's a common belief that if somebody has a brain injury, that you should be able to know it by talking to them or watching them. The key is there are ways to test for it, but they won't always show it in normal human functioning. But we'll find out some key tests where you can show evidence of brain injury. So please be careful not to fall for this myth and disregard someone who says they've had a brain injury and not believe them just because they look and appear normal. Going back to our 10% myth, it's one thing for us to believe in something that's not accurate, but it's the next step, as here in an advertisement, to suggest that there is a way to improve on this false belief, which means that if you're flying this airline, you're not one of these 10%ers. Maybe you're up to the 20%. And if we believe in myths, then we open ourselves up to false claims to be able to fill in the gap, one that we actually don't have. And here's today's key statistic, which is 20%, which refers to the amount of oxygen of the total that we breathe that our brain uses. So first of all, you can see that the brain relative to our body has extremely high energy demands. Also, we'll find out that we can use this as an imaging technique to see which areas of the brain are the hottest or using the most oxygen at a given time or when somebody's performing a given task. Before we get started as well, we want to think about what neurotransmitters can represent. So first of all, we have what's known as the dopamine and the opioid systems. Dopamine is the classic addiction pathway. It is our reward center. It's involved with some other things, but almost any time we are anticipating something. We need our next fix of something, and of course it doesn't have to be specifically chemicals. It can be shopping, it can be sex, it can be that next text or email or call. The key for us, though, is the first time we experience pleasure, whether it's actually activating dopamine and exciting us as a cocaine high would, or the first time, for example, that we take opioids that are downers, such as pain medications or tranquilizers. The first time we take these different substances, they activate different transmitter pathways. However, once we've made that basic association that this thing that I'm doing is associated with pleasure, the brain reorients itself to seek that reward in the future. And what that means is that the first time, for example, somebody has a cigarette, it has some stimulatory effects on the body, but psychologically it might have some relaxing effects. In the future, once that association has been made, the person, when thinking about or sees an advertisement for cigarettes, then their dopamine reward system will become activated. So the key is that for all of our addictions, it seems that this dopamine pathway is what gets trained to light up, to cause us to anticipate and seek that reward, oftentimes overriding our higher order cognitive functions, trying to get us to think of anything else. What this shows us is that once we understand which pathways are involved and which neurotransmitters are important, we can intervene both electrically and chemically to try and deal with the issue. For those of you who have heard of the far side or are far side fans even, I thought this was a good illustration of something that we need to keep in mind when we're talking about our brains and nervous systems. Basically, between species, when we compare one species to another, the size of your brain relative to the size of your body predicts how complex the cognitions that you can show will be. So for example, those with a large brain relative to a smaller body, such as humans, and our closest evolutionary relatives, chimpanzees, will be predicted to be able to do very complex cognitive tasks. On the other hand, dinosaurs with small brains and large bodies probably were not very adept cognitively. Now, we can also try and see if this applies within a species. So within humans, does a bigger brain mean more ability cognitively? And in turn, we find out that that is no. Humans have evolved to have the brain size that we have, and larger brains than average, in fact, are correlated with some psychological disorders and cognitive impairments. So bigger is better between species, but not within our own species. Let's get to some terminology. First, we'll start with some genetic issues. We won't spend a lot of time, and we could, but I wanted to at least introduce some concepts here. First, we have the notion of looking for traits that are influenced by genes. Which genes are the most important ones? As you can see, I've said not the oh God, which in some circles is referred to as one gene, one disorder. Things like Huntington's disease and many physical traits, such as eye, hair color, whether you have a widow's peak, a chin dimple, for example, we can target that to one gene, and that tells us what that person is going to look like, or what, in the case of Huntington's disease, what their brain will look like as an adult. And so weirdly with Huntington's disease, when you're born, you know that you'll have it based on your parents' assortment of genes that they passed on to you. The problem is we don't understand why sometimes it takes until adulthood to start or to kick in. So once again, polygenic means that most of the things that we're concerned with in psychology will not be affected by just one gene. We're looking at many genes. In some cases, for something complex like intelligence, there may be dozens or even hundreds of important genes that we need to consider. Going back to the Huntington's disease example, what causes the gene to express itself at a certain time and not express itself other times? This leads us to the idea of epigenesis, meaning that even though you have a certain genetic configuration, you may be able to turn it on or off. One example that I came up with would be butterfly color. So there's examples of African butterflies that in the wild, at certain times of the year, their wings change a certain color. Transplant those certain butterflies to around our area, in Ohio, for example, and then they do not change to the predictive colors. So what it means is there's something in the environment that the genetic code in the butterflies is responding to turn off or not, to express this gene or not. There's also the example of the oncogene. So for example, I'm not an expert in medicine or cutting edge cancer research, but I know there's two general pathways. Do we target each specific type of cancer? There's reason to do that. There's also a strong suspicion, on the other hand, that most cancers can be linked to a few genes that seem to always be expressed. So if you can find that gene and turn it off, maybe you can greatly reduce one's risk, not just for certain kinds of cancer, but many kinds of cancer, due to the idea of epigenesis. Maybe we can have something in the environment that prevents this gene being expressed once we found it. And this finally leads us to the title of this field. Genetics is exploring the genetic influence, first on how people looked, physical characteristics, but we're expanding it here for psychology into behavioral genetics, linking genetics to behavior, which again, due to this idea of polygenic and epigenesis, makes it much more difficult. So for example, there's an influential book in the 90s called The Nurture Assumption, basically that humans have assumed incorrectly that parenting styles and schools are more influential than they really are. Later research has found out that that is somewhat true, depending on which part of childhood development we are actually talking about. And we'll focus on that question in the future as well. As we continue our exploration of the nervous system, let's start with the basic building block, which is the neuron. As you can see, we have three primary structures of the neuron. First are these dendrites, these branch-like extensions. They connect to the cell body, which does things that a normal cell in the body also does, such as generating energy, cleaning up waste. This large thing attached to the cell body is called the axon. This is what the electrical impulse that the cell body generates when it fires travels down to get to the end and attach to the dendrites of surrounding neurons. Note the flow of information. Information comes from surrounding neurons to the dendrites into the cell body, which generates an electrical impulse that then can be passed in one way or another to the surrounding electrons. Also, we have what's known as myelin, or the myelin sheath. This is essentially an insulation to speed the electrical impulse generated by the cell body. The myelin sheath is typically not present on most neural pathways at birth. However, it does form at some point in childhood. Much of it early on, some of it later on, and as we'll find out, that has very important implications for when certain skills start to develop and develop greater efficiency. Also keep in mind that not every neuron looks exactly the same. Specifically, axon lengths might differ. For example, in the front of the brain, right behind our forehead, the prefrontal cortex, we see lots of neurons packed really closely with very short axons. However, compare that to the nerves in our spinal cord and arms and legs, and they might have axons or bundles of axons of neurons that are a foot or longer, for example. Some disorders can attack the myelin, and as you might imagine, MS, which strips myelin out of select neural pathways, results in more difficulty and slower performance of behavior compared to before the disease was contracted. Let's also look at some related terminology that we'll need to be aware of. First of all, we have neurons in our brain, and our brain is very important for interpreting our environment and directing action, but by itself, it's pretty helpless to do those things. So first of all, it needs to receive sensory information from sensory channels. So this is taking information from our eyes, from our ears, from our fingertips, and relaying that information to our brain. And then also our brain has to relate to our muscle or motor system to be able to generate movement and reactions to our environment. And a key difference between a neuron and a nerve is that a nerve is made up of multiple neuron axons usually. We also have the very simple to ask but very complicated to address question of how does a cell actually fire? We will not dig into this as deep as we could, but it has to do with this idea of polarization, depolarization, and that affects the electrical charge that the cell body can generate. For example, a resting neuron has a negative internal charge with positive charge outside. When it fires, it depolarizes and reverses this. So after it fires, there's a brief refractory period where it cannot fire. However, fairly quickly, within a matter of split seconds, that cell is then repolarized and is ready to fire again. How does this work in even more detail? Well, we'll keep it simple. It has to do with sodium and potassium ions, but those are going in much more detail than we need for our purposes here. Now that we have a better understanding of how individual neurons work and are set up, let's explore how they interact with each other. First of all, we have what's known as an action potential. This is the electrical impulse that travels through the axon. But an important concept to know is that neurons do not actually touch. The dendrites of the receiving and the axons of the sending neuron do not physically touch. They're set up unlike our electrical grid, for example. So what that means is each time a cell fires and generates an electrical impulse, it just represents the potential to actually communicate with the receiving neuron. We also have this notion of a threshold. What this means is that each neuron has a level of stimuli that it needs, activity from the surrounding neurons that causes it to fire, above which it does fire, below the threshold it does not fire. The key concept for us to know is that the thresholds can change over time, and if we're going to learn a new task, it is almost never through growing new neurons. Instead, it is through changing the thresholds of our existing system of connections. So for example, this is a very, very simple neural network. This is maybe the neuron that we want to know whether it's going to fire. Its threshold is too high that this neuron by itself firing will cause it to fire. Maybe this one, since this is a thick arrow, if this one fires, that would be enough stimulus to cross the threshold of this one to fire. And of course, think about with billions of neurons and many more billions of connections between neurons, our neural system is very complicated. In fact, estimates of downloading all the neurons and connections between neurons in one human brain would represent almost all of the known computer storage on Earth as of around 2014. So that means we could maybe represent a brain and a half with all the computer storage out there. So the science fiction idea of downloading our brain onto a computer network is probably not possible at the moment. Of course, when we see actual neurons, this is a stained actual sample, it's really unclear what we're looking at. It's hard to see what are the meaningful wires, what's not. We see this contrast here to represent the cell body, but I believe this is an axon, but even I have no real confidence in what we're looking at. It gets ugly, it's hard to map, and it just helps to appreciate how complicated the brain is. Let's continue our discussion of how neurons communicate with each other, and it has to do with the idea of neurotransmitters. These are chemicals that cross the synaptic gaps or synapses. What is the synapse? As mentioned before, that is the physical space, the physical gap between the sending neuron and the receiving neuron. While this makes the whole process of neurons communicating with each other more difficult, the upside is that if we do want to interfere with this process, now we can use not just electrical stimulation, we can also use chemical stimulation to impede or improve the flow of information between neural pathways. Here we see the process all together. Neurotransmitters are released from the sending neuron's axon, received by the receiving neuron's dendrites. Once the threshold of the cell body is passed, it generates the electrical impulse that travels down the axon and will do so faster if that axon is myelinated. Once that electrical stimulus gets to the axon terminal or end, the electrical impulse dies, but it causes the release of neurotransmitters, and the process continues, and communication between neurons has been achieved. We can also look at different pathways in the brain that are particularly heavy for the activity of certain neurotransmitters. Here we see the dopamine pathway, which represents the reward center, as mentioned earlier. Also keep in mind, as we'll learn later, much of our higher order thought and processing and decision making takes place in our cortex. The reward pathway of dopamine does not have much activity in our cortex, which can help explain why even very smart people become so devastated by addiction. Their higher order thinking cannot override the basic working of the dopamine pathway. Here we see the activity of the serotonin pathways. Compared to the previous slide of dopamine activity, we see that serotonin spreads out in certainly our older, more primal centers of the brain, but also our higher order and newer evolutionary parts of the brain. What this means is that if we're going to interfere with serotonin activity, we know we're going to likely affect a lot of different feelings and thoughts and behaviors. We want to keep in mind that drugs work in different ways. Most commonly, keep in mind that most drugs do not actually give you more of the neurotransmitter in question. Most of them, on the other hand, interfere with the natural rate of activity of that neurotransmitter. So here we see a binding site. The neurotransmitter binds to that site for that specific neurotransmitter. An agonist can block what's known as the reuptake process. So we're agonizing or speeding up the activity of this neurotransmitter. The normal process is that neurotransmitters are released into the synapse, and what isn't absorbed and bind right away gets taken back up to the sending neuron in the process called reuptake. An agonist will block those reuptake pathways, which leads to more of those neurotransmitters being stuck in the synapse, and that causes more activity of that pathway. An antagonist, on the other hand, does not actually bind with the receiving part, but what it does is it prevents the actual neurotransmitter from getting through. So with drugs, we can speed up or slow down the natural activity of the pathway, but we don't change the levels of the neurotransmitter that we started with in most cases. Let's discuss some neurotransmitters that we might revisit throughout the semester. There are over a dozen that we could talk about here, but we'll just stick to the big ones. First, we have dopamine. As mentioned earlier, this is linked to learning, specifically associative learning. For example, being in a certain place or seeing a certain person. Do we associate that with a good feeling or bad feeling? And the dopamine system will reorient us towards seeking that good feeling. As you might imagine, things like cocaine almost exclusively activate the dopamine pathway. And when the dopamine pathway is firing, we experience emotions such as euphoria and ecstasy. This neurotransmitter is also linked to schizophrenia, which is almost universally characterized as overactivity of dopamine pathways, which makes sense that the recommended therapy, among others, is almost always an antipsychotic which slows down dopamine activity. It can work the opposite way for a disorder like Parkinson's, where the difficulty is under stimulation of dopamine pathways. We also have acetylcholine. This is important in the brain, in a very small area of the brain, but hugely important for being able to remember explicit details. What I did yesterday, whose names were the people I hung out with yesterday. In the body, acetylcholine is also important for muscle contraction. When we link that to the characteristic disorder of Alzheimer's, what we find out is that when someone contracts Alzheimer's, plaques and tangles almost universally target the acetylcholine pathways, resulting in memory impairment, and then expand out to other areas of the brain. We also have endorphins. The name is actually very helpful because it is endogenous morphine that's produced in our own bodies. And so, of course, what morphine does is not necessarily give us more endorphins. All it does is cause us to blow out our stash very quickly. And of course, with morphine, you have to keep upping the dose pretty drastically because your body can't replenish that morphine or endorphins as fast as you're using it under the effect of the drug. Also, we can naturally cause the release of endorphins, especially in the case of endurance sports. Relating to the experiences of feeling alert, anxious, and highly aroused and ready to go, we will probably see spikes in our norepinephrine levels. And finally, serotonin, the big one, related to mood, related to depression specifically, also hunger, also body temperature regulation, many things. Now, it might sound interesting and surprising to you that if you're, for example, depressed, you're also much more likely to at some point in your life be diagnosed with an anxiety disorder as well. Depression and anxiety seem to be very different disorders with a lot of important behavioral and cognitive differences, but for some reason, a big underlying similarity is their effect on the serotonin pathways. Here we see all of these neurotransmitters put together. Unlike the previous slide where it seems that one neurotransmitter almost exclusively affects one psychological disorder, in many examples, especially with our mood and anxiety, there's a lot of overlap, which means that medication is not as straightforward as we would like it to be, and also that the effect of the medication can be different across different kinds of people. Let's use some actual examples of how drugs work. Here we're dealing with dopamine pathways. Amphetamines are agonists, so they speed up the process by blocking the reuptake. So normal release of neurotransmitters, but what normally would be sucked back up is forced to remain in the synapse. On the other hand, we have a dopamine antagonist. This is chlorpromazine, a major antipsychotic drug, which blocks the receptors. So even though there is dopamine in these synapses, it all can't get absorbed because there's not enough binding sites. And of course, the next problem is how many of these sites do we need to bind up? What is the proper level of the drug that we want to get to optimal regulation? And usually it's a trial and error exploration. One important thing to remember with all this discussion is that our body typically has a happy, natural way of balancing and regulating our neurotransmitters. The more that we mess with these through things like drugs, the more that it's tough to get them back in line. So the best idea for us to deal with maybe some complications that we're experiencing with abnormal neurotransmitter levels is to do natural things that are linked to getting things back right. And this is usually the obvious stuff, such as getting more sleep, being on a regular schedule, eating healthy foods, and as we find out later on, where do these neurotransmitters come from? In many cases, the gut. And so the food we eat, psychobiotics, can also be linked to having the proper amounts of the neurotransmitters that we need. Let's do a quick breakdown of the nervous system. First of all, we have the brain and spinal cord, which we'll save for another slideshow. Here we're going to focus on the peripheral nervous system. I'll put this tree back up after we've gone through all of these. But first, we can break down the peripheral nervous system. So any neurons in our body that are not in our brain and spinal cord. Some are motor neurons that control our skeletal movement in the skeletal nervous system. Some control our autonomic, automatic sounding features. And we can even further break that down into the sympathetic and the parasympathetic nervous system. So let's explore each of these in a little bit more detail. The whole thing put together is the nervous system, which we break down into the central, CNS, and PNS, peripheral nervous system. A key definition here is that it is our electrochemical communication system, meaning that we need to understand both the electrical aspects and the chemical aspects to intervene if we choose. The central nervous system is simply the brain and the spinal cord. And the PNS consists of the sensory and motor neurons in the body. Here we see where many of our major nerves in the body reside. One problem is that with damage to nerves, for example, that pathway of sending and receiving communication to the brain is damaged. And there's a lot of research trying to figure out how to bypass that bottleneck if it is damaged. When we look at the peripheral nervous system, we break it down into the autonomic, which controls our internal organs. Most of these, of course, we don't really have conscious control over. Sitting in my chair, I can't tell my liver to work harder and expect it to listen to me. And then we have the skeletal nervous system, which is responsible for all of our motor movement. And of course, the skeletal nervous system controls our muscles. Our muscles are attached to our bones, and that's how we produce movement. Further breaking down the autonomic nervous system, we have the sympathetic, which is arousing or energizing, particularly in times of stress. If you've ever been scared at a movie or with a spider jumping on you, you can feel what your sympathetic nervous system kicking in does. Your heart rate increases, your breathing increases. There's going to be some muscle tension, for example. The opposite of that is when we're trying to stay awake. We want to be aroused. We want our sympathetic nervous system to be active, but instead our parasympathetic sometimes kicks in. It slows our heart rate, it slows our breathing, things like that. The key is our body expects both of these to be active at regular parts of a 24-hour cycle. So even though we would like to stay awake all night and all day and just keep the caffeine coming, we need our parasympathetic nervous system to have time to do things such as to replenish our immune system, for example. As you can see here, we have our parasympathetic nervous system with these dotted lines and our sympathetic nervous system with these solid lines. And most of the organs in question are part of each system, which means that fortunately they act automatically and in concert, all signing up to do what they need to do in that moment of stress or in that moment that we relax before bed. What's interesting is if, for example, your sympathetic nervous system, your parasympathetic nervous system is active, which means you're relaxing, you're slowing down, but your heart is racing, that's usually a symptom of some problem if one of your organs happens to be out of sync with the parasympathetic or the sympathetic nervous system. And once again, we can see how all of these systems break down. We will refer to them at different times throughout the semester. And of course, coming up soon will be our extended breakdown of the central nervous system, particularly the brain. One more key statistic. Perhaps you've heard of the case of Terry Schiavo. This was in the 90s due to a lack of oxygen, potentially from an eating disorder related incident. The cerebral cortex was severely damaged and compromised due to this severe lack of oxygen, causing a persistent vegetative state. There were major debates between the family and the husband about whether she should be taken off of life support. Remained on life support for over a decade, even though the professional opinion was there was really no expectation that any of the previous higher order abilities of Terry previously would ever come back. So a physically excruciating tennis match or other endurance event, even though your body should be breaking down, you don't feel the pain. What is this linked to? It is your body kicking out endorphins. So let's not misinterpret this. Your body is still in pain. It's hurting. It's just that your brain is not receiving those signals. So even though you're feeling great, don't let that mislead you into thinking that your body is also feeling great as well. It's just an emotional, psychological state. But it is nice to have, I will say that. White matter, what would that sound like in terms of the different parts of the neuron that we covered? And it comes down to gray matter is what we start off with in most of our brain. Those pathways that we are actually using, the brain seems to realize are important because we're using them and myelinates them, turning them white. We've all been here. We've all heard something embarrassing about something we've done, and we blush. What is this activating? We weren't trying to do it. It was an automatic function, so it is the result of the autonomic nervous system. Let's see how we're doing with our understanding of neurotransmitters. Overactivity of this is linked with schizophrenia, dopamine. Key pathways in the hippocampus, which we'll later find out, is highly important for memory. This would be acetylcholine pathways. Pain medications almost universally affect endorphin levels, opioid-based especially. The neurotransmitter most commonly linked with addiction, as we discussed, is dopamine. Not just chemical dependency, but many behavioral addictions as well. The neurotransmitter that we would expect to spike in stressful situations will be norepinephrine. Related to hunger regulation is serotonin. One example of this being a problem is that many drugs that treat depression are called SSRIs, or selective serotonin reuptake inhibitors. What they do is prevent the reuptake of serotonin, keeping more in the neural pathways and causing those pathways to become more active after taking the drug. However, as you'll know, if you've taken medications, there are many side effects. Since serotonin affects things such as hunger regulation, temperature regulation beyond mood, you might see effects on those things that you wish the drug didn't affect. Final true and false for review. This would be true. If you think of the difference between a 3-year-old and a 6-year-old picking up a pencil and writing their name, a lot of that increased efficiency is not just due to physical practice, it's due to how the brain represents those pathways, and myelin has a big factor in that. This is false. The sympathetic nervous system is part of the autonomic nervous system. Dopamine overactivity, as we discussed earlier, schizophrenia. So this would be false. Epigenesis involves whether genes are expressed or not. This is absolutely true. Also, organs can be part of both the sympathetic and parasympathetic. It's important that they're quickly mobilized to do different things based on different situations we face, arousing or relaxing. We also have the idea of damage. What does this mean? This is a bit of a preview for what we'll talk about in the brain slideshow, but this is false. If we think about Terry Schiavo, which we mentioned before, she was referred to as being in a vegetative state. That is much different from somebody who's had a prefrontal lobotomy. They can still move around, they can still walk and talk to some extent, but specifically with the prefrontal lobotomy damage, you've lost a lot of your human motivation and judgment and interest in other humans. But it is different from a vegetative state. For our key study, I wanted to introduce you to one of the big names in psychology that may not get a lot of attention historically or in the textbook, but this is Paul Ekman. He is known as the emotions guy. He has studied emotions across the planet, looked for cultural similarities. What are the universal emotions? Many of which he explored in this particular study. He has also, interestingly, spent a lot of time to control the muscles in his face. Most of us are not good at, for example, generating a true, genuine smile. Paul Ekman can do it through a lot of practice and understanding of what constitutes a true, genuine smile. For this study, however, what he was looking for was to challenge the common assumption that first we have a reaction to something, we like it, we don't like it, and then our body responds. We smile, we frown. What Ekman wanted to show was if we turn that causal association around, what happens? If we just train people to smile in a genuine way or express other emotions through facial expressions, what then happens to our internal systems? Does it affect our autonomic nervous systems? And what we found is that here are some of the primary emotions explored: anger, disgust, fear, happiness, sadness, and surprise. When we make people do these faces, we see changes in autonomic functions such as heart rate, skin conduction, finger temperature, and other indicators of body activity. Let's look specifically. We can see the abbreviations for the different emotions, and we can see, for example, we're looking at heart rate here, and we see that someone making an angry facial expression causes their heart rate to spike. Interestingly, showing surprise does not change their heart rate. We also see muscle activity. So when, for example, we make a face that corresponds with happy, we find out that we see more muscle activity compared to the change in others. So let's discuss some of the concepts we learned about biopsychology. How does this fit with some of your own experiences? How can we apply some of what we know about biopsychology to improve our lives or to understand the lives of those around us? Also introduced would be the use of drugs, or we can also talk about options for electrical stimulation in the brain that we'll cover at other times in the semester. What is the status for the legal or moral or ethical debates about drugs in our society and how we use them, and when should we be allowed to and when should we not be allowed to use them? So looking to discuss all of this and more in the class discussions.